After going through the processes of transcription and translation, we move into how we control the process of gene expression. We'll cover prokaryotes and eukaryotes because regulation of gene expression is a bit different between the two, as are many processes within our cells. So why do we need to control gene expression? Well, the very simple reason is that not all genes need to be expressed all the time. And there are several reasons for this. One is that the environment changes. So particularly for prokaryotes, when their environment changes, they have to change their cellular response to that environment. And so this might mean that a nutrient changes, something is lacking or something is present that isn't normally present. And so the prokaryote has to respond by changing the type of genes that it expresses. Now, this could also be changes based on interactions with other organisms. And it could also be pretty much anything to do with a shift in any nutrient, change in temperature, anything like that in the environment. Gene expression also changes because cells are specialized in eukaryotes. So obviously this doesn't matter for prokaryotes, but for eukaryotes, each cell type has its own structure and function, and that is related to the genes that it expresses. So if a cell doesn't need a particular gene because that's not part of the function or the structure of that cell, then it simply doesn't need to be expressed. Expressing genes takes a lot of energy, especially the process of translation where we're building these long chains of amino acids. And so we're not going to produce things that we don't need. And then also for eukaryotes, different times during growth or development, we need different genes turned on. So this might be related to hormones, it might be related to changes in cell types or proportions of cell types. So the cells need a way to control not only when a gene is expressed, but how much of that gene product is expressed when it is turned on. And both of these can be regulating factors for controlling gene expression. We're not really going to talk about controlling the amount of gene expression. We're going to talk about the very basic ways that genes are turned on and off, but they can also be regulated by how much. So one of the indicators for turning a gene off can be that there's plenty of product. There are several types of genes that we might find in a genome. The main type of gene that we're going to find are structural genes. These are our genes that encode a messenger RNA and the instructions for a functioning protein. On top of that, we have a suite of genes and elements whose function is to regulate those structural genes when they turn on or off. So we have some genes who are essentially structural genes whose function is to regulate transcription or translation. So the end product of these genes actually has some sort of involvement in whether or not transcription or translation continues. And then we also have regulatory elements. These are DNA sequences that are not transcribed, but are playing an important role in regulation. So these tend to be structural changes. We'll talk about something called epigenetics a little bit later, which is a structural change, usually by methylation, that regulate whether or not a gene is expressed. So we don't regulate whether or not a protein is expressed, we regulate the structure of the DNA to affect whether or not anything can be transcribed. We do see some differences between prokaryotes and eukaryotes with respect to both their gene structure and the ways in which they are regulated. In bacteria, most of the regulation happens at the point of transcription. So if the gene product is not used or not needed, then it won't even be transcribed. Whereas in multicellular organisms, we tend to have more extensive types of gene regulation. So in bacteria, the goal of gene regulation is to maintain a sort of standard environment inside the cell. And so they have to be able to respond very quickly to changes in the environment. So bacteria can work to change the expression on or off of their genes very, very rapidly. Now, that's not to say that eukaryotic organisms don't do this rapidly as well, but they do have a more variety in the mechanisms that they can use. In eukaryotic cells, not only do we have the regulation of what gets expressed when and where, but we also have regulation of genes that control the type of cell that we have. So there's extensive regulation for cell differentiation to make sure that we have not only the proper genes that are being expressed, but also the proper cell types that the organism needs. As we continue through our discussion of prokaryotes and eukaryotes, we're going to talk about some examples of how genes might be regulated. In order to get there, we need to talk about the general mechanism in which genes are regulated. So a constitutive gene is one that is turned on all the time under normal conditions. So we call this constitutive expression. And it basically means that the default for gene expression is on. And so until it is turned off, those genes are just going to continue to be expressed. Some genes are under what we call positive control. Positive control means that regulatory elements are going to either turn on or increase gene expression. So sometimes we might need to stimulate and increase the expression. Other times we might need to just turn on the expression in general. A gene that is under negative control means that it either needs to be decreased or turned off altogether. So we're going to talk about some specific types of positive and negative control, specifically during our prokaryote discussion. So we'll come back to this a little bit later. Gene expression can be controlled at many different places along the process of gene expression. For this video, I'm going to go ahead and just go through and talk about them, and then I will let you pause to write down all of the different steps. So the first level of gene expression that we can look at is alteration of structure. So remember that during cell division, we talked about how some chromosomes are more relaxed. This is called euchromatin, and other chromosomes are more densely packed. This is called heterochromatin. So the very first level at which we can regulate our expression is to change the structure of the DNA. And there's a couple different ways that we can do that. We'll talk about one a little bit later. But basically, by controlling how tightly packed a gene is, we can control whether or not it's expressed. So if it's hard for cellular machinery to get to tightly packed DNA, then that DNA is not going to be expressed. Likewise, you can make structural changes to the DNA to keep it from being expressed. And we talked about this a little bit ago, or I mentioned it anyway, that this is something like methylation, where we add a methyl group. And what this does is it just kind of blocks RNA transcription factors from actually binding to the DNA so that they can't build anything. We can also regulate at the process of transcription at the point of pre-RNA, mRNA processing. We can also regulate the mRNA molecule itself. Remember that we added these caps and tails onto our messenger RNA as a way to help stabilize the molecule. So if we remove that cap or that tail, then we're going to lose the messenger RNA because it will be degraded. And so we can regulate just there right before translation as well. You can also regulate at the point of translation and protein activation. So once a protein is produced, it has to be completed and activated in order for it to work. So if translation can be regulated at the point of finishing up that final protein. And then finally, we also can have post-translational modifications. These are modifications that happen to the physical structure of the protein that keep them from functioning or cause them to function as needed.